Looks like we're still waiting for one committee member.
Yeah, Brenda's here, Mark is here.
I don't see Brian yet.
Brian is, yeah, the one I haven't seen.
Okay. Still waiting on one person. I know he knows because he just emailed not long ago.
Okay.
There he is.
Cool.
Okay, I think we have all committee members present. So we shall launch in here. And well, thank you all for coming during a very busy time of the year, I know, and for joining June for her master's defense on this very auspicious day, which is the first day of June. And of course, in true June fashion, she chose this day specifically for that reason. Gosh, I don't even really know where to start with this intro. June is such an integral part of our lab. I've actually known June for 9.5 years, which is actually not quite true. She's known me for 9.5 years. I would say I've known her for more like 7.5 years. The reason there's a difference there is she was in my ecology course way back in the fall of 2013 when she was an undergrad. And that's a big course, so I honestly don't remember her. So sorry, June. But when I really started to get to know June was when she enrolled in my much smaller, more discussion-based biogeography course 2 years later. And then I feel like I really, really got to know her starting in the spring of 2016, as we were, elbows deep in mud dealing with the crumbling remains of the Reeser mammoth. So she worked really long and hard hours to stabilize specimens. If I remember correctly, she had this giant piece of the pelvis that was like too big to even move. And so she was an extremely dedicated mammoth worker that spring in paleobiology, and her excitement and willingness to go above and beyond into the muck to deal with that find throughout that whole arduous time was her excitement was palpable. And in fact, I can't fully remember, but I think she gave the OSU mammoth its nickname, Crumbles, which is what we still use today to refer to it. So if we rewind back to pre-Crumbles time, the June I first met was really shy, really quiet, but she got up the nerve to ask me if I had any volunteer positions in the lab. And it was an interesting time to be asked. I was extremely overrun with morning sickness at the time. Nobody knew, but I was throwing up in trash cans after every class. And so you know what I said to her? I said, no, unfortunately, I just couldn't take anyone else on. And oof, what a mistake that was. Luckily, June was extremely persistent and she didn't agree with my answer. And so she kept asking, and she asked several more times. And soon after the whole Crumbles experience, my grad student, David Taylor, and I needed someone that we could trust to image fossil mouse mandibles to try out some morphometric analyses to see if we could set that up in our lab. Now, imaging tiny little mouse mandibles is about as far a cry as you can get from, working with gigantic mammoth remains, but June was game. And so that was it. She quickly then became an indispensable part of the lab from then on out. And I'm just so incredibly lucky that she didn't take my initial misguided no as an answer, because June is such a gem and it's been such a privilege to have her in the lab. So after she graduated, she actually stayed in the lab, thankfully, as a post-bac lab assistant after graduation. And she even took on a lab manager role for a time, and then decided that she wanted to actually take the plunge into grad school to expand on the work that she had started as a lab tech, actually expand it into her master's thesis. So I'm so intensely proud of the scholar that June has become, and the work that she's produced for her master's. I'm excited for you all to get to hear about it. She has poured herself into this work, into this journey in grad school, and she's transformed into this unstoppable student and scientist. And on top of all of that, of course, the pandemic hit early on in June's time as a master's student. So, she completed her master's with the added difficulties imposed by the pandemic, including lab and museum shutdowns that totally snarled her plans early on in terms of collecting data. And of course, all the isolation that resulted in as well. And then she also has had to deal with an advisor who had to drop everything and go on medical leave to deal with 2 family medical emergencies last spring, and who's been gone on sabbatical, still dealing with the fallout from that this year. So June, you are such a rock star. Thank you for being so patient when everything felt like it was on fire. And I'm just so grateful to you for everything these past 7.5 years. It's been an honor to work with you through all the different iterations of your time at OSU. I'll close by saying, June has the best wry sense of humor ever. Even when times are tough, she never fails to make us laugh. She's also just ever supportive of those around her. She cares very deeply about people, be it her family, her lab mates, her friends, her students, her dogs, of course, even the cows on the farm where she lives. She's actually attended the births of several calves, I think, at this point during grad school. So there's a skill that she has learned, not in the Terry lab, but still, deals with mammals. So in addition to delivering livestock, she also she's a very talented artist. She's a master at homemade donuts. So before the pandemic, she would treat us to homemade donuts. And let's see, she introduced us all to, I'm going to butcher this word, gjetost, a Norwegian cheese. And she actually lent my kids one of her prized collections of comics, the Moomin comics. So she's just, expanded horizons in all sorts of different ways for all sorts of different people associated with our lab. So if you don't know those comics, go look them up, Moomin. And the last thing I'll say is I want to give Dirk a special shout out because of Dirk and her other faithful 4-legged companions who have been on this journey with her, but especially Dirk. Our lab's unofficial mascot has been the dachshund for all of these years, which is kind of ironic if you think about it, because we do mostly study small mammals. So thank you, June, for bringing dachshunds into our lives and for all that you have taught me personally and us in the lab about just, how to tackle life's challenges and move through life with grace, with dignity, with honesty, perspective, humor, and just enjoyment. So it's my utmost pleasure to hand Zoom over to you now so you can present your master's research. So take it away and good luck.
Well, thank you so much, Becca, for that intro. And thank you guys for all attending. My name is June, and this is my defense. The long and short of it, dynamic ways in which small mammals respond to environmental gradients in time and space. It's a bit of a mouthful once I say it out loud. So there we go. Okay. Broadly, this thesis is going to attempt to better understand the ways in which species are responding to environmental change. I tackle this by using small mammals in the Great Basin I combine fossil data with modern spatial data to assess how species are responding across temporal and spatial scales. This will serve as my roadmap as I walk you through my research. I want to start by asking, what are environmental gradients? Put simply, these are changes in abiotic factors across a spatial or a temporal scale. Factors such as temperature, precipitation, plant cover, and many more play a vital role in establishing these large-scale gradients. Species are often tied directly to these environmental gradients, so when a major change occurs within one, species are forced to respond. When species respond to these large-scale environmental changes, we can categorize their response into one of three ways. They can move, often in the form of migration, range shifts, expansions, or contractions. Additionally, they can adapt or acclimate. And this is to say that they remain in place and respond directly to the environmental change around them. And lastly, if they can't move, they can't adapt or acclimate, in time, they're going to die or go extinct. My thesis focuses on this middle response, adapting and acclimating. So what does it mean to adapt or acclimate? Adaptation is a form of long-term change shaped by natural selection, sometimes referred to as evolutionary change. To the right, we can see the chiseled incisors of Dipodomys microps, a highly specialized kangaroo rat who has co-evolved a unique mandibular and incisor shape to exploit salt off of Atriplex, a type of desert salt brush. This is a form of a morphological adaptation. On the other hand, we have acclimating. And acclimating is a short-term change spurred by an immediate need, sometimes called a plastic or a behavioral response. Great example of acclimating is diet. Diet can fluctuate depending on what is available, and while it isn't permanent, it may provide a species with enough buffering to allow themselves to thrive in the face of environmental and ecological change. Both of these examples are correspondent to diet, but vary in their nature. The former is a type of tracking and specializing on a specific resource, while the latter is a response in the moment to environmental change. In this thesis, I set out to uncover the ways in which species are responding in situ or in place. Is their response rooted in adapting or acclimating? Is it evolutionary or plastic? I also want to tease apart the relationship between these axes of variation, morphological and dietary, and see how those are coupled or decoupled across time and space. And lastly, I want to set out to assess how interchangeable spatial and temporal scales are, and whether or not substituting space for time is applicable, and if so, what can the combination of these two scales tell us about species' future response? For questions like these, a lot of species can provide answers, but characteristics of a taxa that make these questions easier to answer are things like fast generation times, known levels of adaptation and plasticity, diverse communities, and high levels of co-occurrence. Small mammals satisfy these needs and many more, and serve as an excellent model organism for environmental change. Small mammals are what we call bellwethers of ecological and climatic change. Oftentimes primary consumers, small mammals are some of the first to illustrate changes in the resource base and shift in abundance and distribution accordingly. In this thesis, I've chosen 10 small mammals spanning a range of phylogenetic histories, body sizes, and dietary strategies in an attempt to better understand what it is exactly that's helping species buffer themselves in the face of environmental change. I have species from the Dipodidae, Chryseidae, and Heteromyidae families. These are three very diverse families, and Heteromyids in particular represent a highly specialized family adept for very dry and warm conditions, such as the Great Basin. Additionally, I have sorted by body size, with small-bodied species being under 25 grams and the large-bodied species being 50 grams up to our largest species, Neotoma lepida, the bushy-tailed wood rat, at around 350 grams. And lastly, I've grouped species by dietary strategy, with omnivores, herbivores, and granivores serving as these three broad groupings. I've chosen the Great Basin to help answer my research questions, given its high levels of small mammal diversity, as well as broad temporal and spatial scales. The Great Basin is an expansive high desert in the intermontane West. It has a rich fossil history, as well as hosts a wealth of scientific trapping and museum collections. Furthermore, it's highly impacted by climate and anthropogenic change. To the right, you can see a map of the region with the 4 study localities I use labeled. Two-Ledges Chamber and Homestead Cave are 2 caves, are fossil sites spanning the last 13,000 years. These sites will be used to look at small mammal response through time. And in between these sites, I have the Ruby and Toyabe Mountains, two mountains spanning diverse ecological and habitat gradients. These two mountains will be used to look at small mammal response across a spatial scale. And I'll talk more about these individual localities in detail later on. So the Great Basin is part of the Basin and Range Province. It was formed as a consequence of rifting, or simply continental crust extension, around 25 to 20 million years ago. Mantle underflow from the asthenosphere pushed the landscape into a dome-like shape, eventually cracking and creating these north-south trending faults. The buoyancy of that warm underflow from the asthenosphere, combined with the continued thinning crust, created upthrust edges, ultimately becoming the mountains, and downward edges sinking into what was now the valleys. The consequence is this distinctive pattern of basin and range we see across the landscape today, each mountain complete with a strong ecological and habitat gradient across its elevation. The Two Ledges Chamber and Homestead fossil sites, these two caves, are nestled within the Lahontan and Bonneville basins, respectively. These two basins were filled with massive pluvial lakes about 20,000 years ago. With no outlet to the ocean, the waves of these lakes would eventually hollow out caves on these mountainsides across the Great Basin. The area would see warming and drying trends, displayed here by oxygen stable isotope values. Higher delta 18 oxygen values indicate warmer and drier conditions. And you can see that over about the last 15,000 years, there's been a very clear trend towards warming and drying that persists across the Great Basin. As the region is warmed and dried, these massive pluvial lakes have dried with it. The caves they formed became homes for raptors, where they could cough up pellets and in the process, create a wealth of small mammal fossil data spanning thousands of years and covering a strong environmental gradient of warming and drying. Like that temporal scale, modern spatial scales provide an excellent way to measure a species response to environmental gradients. Unfortunately, the Great Basin is full of these mountains, each with their own strong ecological and habitat gradient, and each capturing a species climactic envelope. Across these mountains, we see warmer and drier conditions at lower elevations. Shrubs and grasses dominate the landscape, and increased levels of anthropogenic activity and invasive species persist. These low elevations share a lot of similarities with the modern conditions seen from that previous temporal scale: warmer and drier conditions and invasive species and things like that. As we move up the mountain and increase in elevation, we see pinyon pine and juniper start to take hold, followed by lush grass and willows at mid elevations. At mid elevations, plant and animal biodiversity is at its highest. This is what we refer to as the mid elevation peak. This peak occurs around 2,100 meters for both the Ruby and Toyabe mountains. As we continue to increase in elevation, we start to see plant and animal diversity levels decrease a bit as conditions get colder and wetter, ultimately resulting in the alpine zone with very little plant cover and very cold and wet conditions. As we increase spatially or elevationally, we see that conditions get colder and wetter, similar to how when we went back in time, we see that conditions are colder and wetter about 20,000 years ago with all of those pluvial lakes and storms in the Great Basin. One of the questions I'm interested in is if spatial variations seen today can be used as a proxy for the variation a species could exhibit through time. And one of the nice things about utilizing the fossil record is I can actually check. So I can see if a species response to environmental gradients today in areas of warm or areas of cold mirrors what we've seen throughout the fossil record. It tells me that the variation a species could exhibit spatially today is comparable to that variation they could exhibit temporally. This strongly supports being able to predict species response in the future to even greater warming and drying trends. If it doesn't, however, that means we probably should err on the side of caution when making these blanket space for time substitutions and really understand the local history more when predicting species future response. I've chosen to track the mandible as a means of measuring adaptation. The mandible is the primary skeletal unit used when feeding, thus its form is directly related to function. Small mammal mandibles show a wide range of morphologies with high levels of specialization and generalization. Below, we have the mandibles of the generalist species, Peromyscus maniculatus, as well as the highly specialized Dipodomys microps. And you can just see how different these mandible shapes are depending on what they are eating and their lifestyles. In addition to its biological usefulness, rodent mandibles are two-dimensional, easy to image, and well preserved in the fossil record. One of the two responses I'm interested in is adaptation. And to quantify a species adaptation, I utilize geometric morphometric analysis of the mandible. Geometric morphometrics allows for measurements of size and shape trends. Additionally, it allows for an estimation of degrees of specialization. And it also can tell us about what is driving morphological variation. And using the mandible, this process of geometric morphometrics is really quite simple. We start with imaging our mandible. That photo on the bottom left is exactly that. This is a pretty straightforward process given the Terry Labs microscopic camera. We lay these things down, they're relatively flat, and take an image. Make sure to account for scale for later analysis. From here, I place landmarks on homologous features. I've specifically chosen features that are easy to locate across taxa as well as biologically useful. The landmarks are shown here on this stylized mandible to the right. They dot the perimeter of the mandible with an emphasis on sites of muscular attachment and regions of mechanical advantage. In features, if features are missing, which is often the case for fossil specimens, these things are thousands of years old, they've been partially digested, they've been coughed up as owl pellets. A lot of times things are prone to breakage, especially regions like the angular and coronoid process. So in the case that those are missing, we need to estimate their placement. And to do that, I use a known placement from a reference specimen, a specimen that has all 13 of its landmarks. And the reason we do this, one, is to have an idea of where that would fall, but also if we don't estimate it, all of our numbers are going to shift down, which is going to allow for comparisons of landmarks to get off kilter. Once all landmarks have been accounted for, I superimpose the mandible images, and this is going to account for magnification and rotational differences among images. This way, all that I'm left with is the size and shape difference between images. And from here, I can begin analysis. As I analyze these data, I make the distinction between size and shape. Size is just the general scale of the mandible. It's going to be measured by centroid size, which is the sum square root of points to a centroid figure. This is going to be a proxy for specimen body size. And there's obviously better ways to measure body size, but given the limitations of the fossil record, this provides an invaluable way to assess species body size through time. We don't always have these critters right in front of us, right? If we just have certain bones. And a lot of times even things like molars, which are a great way to measure body size, are not present due to high levels of indentulation. Conversely, shape is going to be what remains when location, scale, and rotation have all been removed. And we're just looking at the difference in placement of landmark coordinates from one image to another. This is going to be measured via principal component analysis of these landmark coordinates. In addition to adaptation, I'm interested in a species' short-term plastic response or their acclimation level. And to do this, I utilize stable isotope analysis. Stable isotope analysis of carbon and nitrogen is going to allow us to estimate a species' dietary niche, size, and position. This isotopic data comes from David Taylor's dissertation and spans both temporal and spatial gradients of interest. Isotopic niche breadth is going to be how I quantify a species' diet. It's comprised of carbon and nitrogen stable isotopes. Stable isotopes do not decay over time, which makes them a great way to measure a species, get a snapshot of a species' diet in the modern as well as the fossil record. Along the x-axis, we have carbon. And what carbon is going to tell us is the primary producer's photosynthetic pathway. We can see that C3 plants are on the left side of this, indicating a more negative carbon signature, and C4 plants are on the right side of this, indicating a less negative carbon signature. It's worth noting that I have accounted for the Suess effect. The Suess effect is just a change in carbon isotopes in the atmosphere due to the increased presence of burning fossil fuels in recent years. On the y-axis, we have nitrogen. Nitrogen is going to give us an insight into the relative trophic position of a specimen, with herbivores and granivores displaying lower nitrogen signatures and omnivores and carnivores displaying higher nitrogen signatures respectively. Sorry. When combined, the isotopic data from a group of individual specimens can give an estimate to a species' broad dietary habits and dietary niche breadth. Ellipses were created for both morphological and isotopic data using the CyBER package in R. CYBER is a package used for isotopic niche breadth, where you will plug in carbon and nitrogen values and start to create ellipses and things like that. I used this program to input principal component scores from the first and second axes of my principal component analysis. This was to allow for a morphological niche breadth that mirrors the isotopic niche breadth created from carbon and nitrogen stable isotopes. The ellipses created from this principal component analysis are going to estimate a species' morphological shape variation, while isotopic niche breadth ellipse area is going to estimate dietary flexibility. Ellipse areas for each of these measures is going to directly relate to increased levels of variation. In the upper right-hand plot, we have that principal component analysis ellipses created from first and second PCA axes. The cyan triangles are going to represent fossil specimens, while the tan circles represent modern specimens. To the right, we have box plots that were created measuring the ellipse area from each of these two ellipses. In the box plots, the largest and lightest colored box in the back represents a 95% prediction interval. The next smallest box in the middle represents a 75% prediction interval, and the smallest, darkest colored box represents a 50% prediction interval. The black dot represents the mode, and the red X represents a 95% prediction interval rooted in 40% of the data. This red X is done to account for small sample size and to minimize the effects of outliers. And from what we can see from this is that the fossil ellipse is a lot larger and thus more variable morphologically than the modern ellipse area. The same principle holds true for our isotopic ellipses below. And this idea of using ellipse area as a measure of variation is imperative to understanding how these two axes of variation are coupled and decoupled in species through time and across space. Moving into my first chapter, I used Two Ledges Chamber and Homestead Cave, two fossil cave sites in the Great Basin, to help answer my research questions. I utilized geometric morphometrics to track morphological trends and stable isotope analysis to track isotopic niche breadths across the last 13,000 years in an attempt to better understand how these small mammals are buffering themselves in the face of environmental change. Two Ledges Chamber and Homestead Cave provide stark contrasts in terms of local history. Two Ledges Chamber is a remote sagebrush-dominated cave in northwest Nevada. It has little anthropogenic impact. And conversely, Homestead Cave is a highly altered cave. It's dominated by things like cheatgrass and other invasive species and has high levels of human impact. This is a military munitions site just west of Salt Lake City, so it's much more altered than Two Ledges Chamber. Specimens from this study come from owl pellets. Two Ledges Chamber data spans most of the Holocene, up about 9,000 years, and comes from the Terry Lab. The specimens here were excavated by Brian Hockett in 1991 and later identified by Rebecca Terry. And Homestead data spans the entire Holocene and into the late Pleistocene, which is about up to about 13,000 years. These specimens came from the University of Utah Natural History Museum, and they were excavated by David Madsen and Don Grayson and later identified by Rebecca Terry. Given that these two sites have different time spans and specimens were dated at different intervals, I felt it was appropriate to assign bins of time periods to allow for cross-site comparisons as well as to visualize broad trends through time. These time periods are listed below, and Two Ledges Chamber is going to span from the modern to about 7,800 years is the oldest that we've dated the fossils to. That's going to be the modern late Holocene, middle Holocene, and the very tail end of the early Holocene. Whereas Homestead Cave goes back a little further. It's going to be from the late Pleistocene across the Holocene and into the modern. I used 9 of the 10 focal species mentioned earlier. These species are found across the fossil sites across the fossil records at both sites, and they represent a wide range of phylogenetic histories, dietary strategies, and body sizes. Zapus princeps is not included here given its absence from the fossil record at both sites. Species listed, these species all listed have morphological data from at least one of the 2 cave sites, and most times at both sites. These highlighted species, Neotoma lepida, Dipodomys microps, Chaetodipus formosus, Paragnathus longimembris, and Paramiscus maniculatus, represent 5 subsetted species that have both morphological data and isotopic data from Two Ledges Chamber. When I talk later about the relationship between morphological variation and isotopic flexibility, I'm specifically talking about these 5 subsetted species. These species also span a wide range of phylogenetic histories. We have heteromyids and chryseidids. We have both large and small-bodied species, as well as omnivores, granivores, and herbivores. I track trends in centroid size for these focal species through time. And here you can see those trends broken down on a species level. On the x-axis, we have years before present, with deeper time on the left side of each individual species graph and the modern on the right side. The y-axis, we have centroid size measure. Salmon-colored points represent Homestead cave data, and cyan-colored points represent Two-Ledges chamber data. The lines represent a localized regression that has been smoothed with a 95% confidence interval. This is here to help visualize trends through time. And what I found is that species are responding rather individualistically across time. There's not a consistent trend across the 2 sites, nor is there consistent trends across phylogenetic, body size, or dietary groupings. Even species that look like they might have some significant trends, we start to see upon closer look that there's a lot of overlap and that a lot of the trends that we see are estimated between points. I did find an interesting case of some possible interspecific buffering. Dipodomys miriami and Chaetodipus formosus are 2 known competitors. Dipodomys miriami is the smallest of our kangaroo rat species, and Chaetodipus formosus is one of the larger of our pocket mice species. And what we can see here from the figure looking at centroid size through time is that these 2 species appear to have an inverse trend in centroid size. And this may be a strategy to help better isolate themselves from direct competition with one another. Dipodomys miriami exhibiting a concave uptrend in that kind of lighter colored brown, and Chaetodipus formosus showing a concave downtrend. Next, I look at shape and how it varies through time. I conducted a principal component analysis of landmark coordinates for each site. Open circles represent fossil specimens. Each cluster of fossil specimens has been grouped at the species level and fitted with an ellipse. Filled circles are going to represent average mandible shape from a given time period. So those are those time bins that I talked about earlier, the late, early, and middle Holocene and the late Pleistocene. Filled in squares are going to be the modern average shape. These have not been included in the ellipse calculation and instead have been included as separate points. So they should not be factoring into the ellipse area. There's been arrows going from deeper time into the modern to show trends in morphospace through time. And what I found is that mandibular shape corresponds really well to evolutionary history and taxa. There's a clear split between chryseidids and heteromyids at both sites, with chryseidids clustering on the left side of the graph while heteromyids are on the right side. The first axis also differentiates between species with larger ramus and elongated condyle processes, and the left side, and the right side is going to differentiate between species with more robust alveolar regions and more elongated angular processes. For most species, their modern mandible shape fell within the fossil range. That's indicated by that filled in square. And for most species, that's falling in that ellipse. However, one notable exception was Dipodomys microps, seen here. We can see that its modern mandible shape fell outside of its fossil range. Across the principal component analyses, kangaroo rats had a shift towards a mandible shape with increased alveolar robustness and an increase to the molar region. Alveolar robustness and molar robustness alike are going to indicate an increased reliance on mastication and chewing of tough materials. In the upper right, we can see a mesh warp of landmark coordinates for Dipodomys microps' modern mandible shape. And what this is showing is it's the modern mandible shape's landmark coordinate placement in relation to its average fossil shape. So anywhere where you see warping on that grid, that's going to indicate high levels of variation from the fossil shape. Below that, we have kind of a stylized version of that same image, looking at where the landmarks are drifting to in the modern. So the filled in circles represent the average shape from the fossil record for Dipodomys microps, and the arrows are pointing towards its new novel mandible shape. You can see there's a lot of movement around the molar region, the alveolar region, and things like the coronoid process are decreasing in elongation and the condyle are widening a bit. Here we can see the ellipse area corresponding to mandibular shape variation at both Two-Ledges Chamber and Homestead Cave. And within each of these groupings, it's been split between the fossil record and the modern. So we're looking at how variable their morphological niche space is. And what we can see is that at Two-Ledges Chamber, kangaroo rats and our generalist Peromyscus maniculatus, there's a lot more morphological variation than there was in the fossil record. Additionally, we see heightened levels of morphological variation at Homestead Cave for Peromyscus maniculatus as well. Something also worth noting is that across the board, fossil and modern, we see higher levels of morphological variation at Two-Ledges Chamber than at Homestead Cave. To assess dietary flexibility, I measured isotopic niche breadth in our 5 focal species. In the graphs on the right, we have isotopic niche breadths from the fossil and modern, grouped at a species level. Fossil isotopic niche breadth is shown by the cyan triangles, and modern isotopic niche is shown by the tan circles. For our 2 large-bodied species, Neotoma lepida and Dipodomys microps, we can see that there's not a whole lot of movement and that the modern isotopic niche is still relatively contained within the fossil niche. However, for our small-bodied species, Chaetodipus formosus, Paragnathus longimembris, and Peromyscus maniculatus, we see major changes in modern isotopic niche. Chaetodipus formosus here in the top right shows almost a novel diet in the modern with much lower levels of carbon and nitrogen signatures, whereas Paragnathus longimembris and Peromyscus maniculatus both show broadening trends in the modern as well as relative higher trophic positions. By combining the morphological variation and isotopic niche breadth as a measure of ellipse area, we can assess how these species are responding through time. Are these species responses coupled or decoupled? And because we're using this ellipse analyses, we can combine these to see if a relationship persists across strata. When I combined morphological variation with isotopic niche breadth variation or dietary flexibility, we see that a significant trend emerges across species as a whole, with increased levels of dietary flexibility being tightly related to increased levels of morphological variation. We also see that this trend is significant within heteromyids, and this is going to support a many-to-many mapping, where many different resources are going to equate to many different morphologies to satisfy all of those different resources. That concludes the first chapter. So moving into the second chapter, I look at the same questions, but I use the Ruby and Toyabe mountains across our spatial gradients. I utilize the same methods from that first chapter, specifically geometric morphometrics, stable isotopes, and then comparing the relationship between the two. Due to species capture data spanning different elevations from different mountains, I elected to bin data by 250-meter bands. I created 7 250-meter bands spanning the focal species captured elevational range and spanning a broad environmental gradient. I used 9 of the 10 focal species laid out initially, with Chaetodipus formosus being omitted due to its absence from the mountain range, and Zapus princeps instead being included, given its high levels of abundance at both sites. These highlighted species represent species with both morphological and isotopic data. Again, I measured trends in centroid size. This time, however, our x-axis is elevation in meters, and that's going to be increasing as we move to the right for each species graph. Cyan points represent the Toyabe Mountains, whereas the salmon-colored points represent the Ruby Mountains. Again, the lines are local regressions and smoothed with a 95% confidence interval to help visualize trends across elevation. Similar to chapter 1, we see that species are really responding individualistically in regards to size, and that there's no consistent trends across phylogenetic groupings, body sizes, dietary strategies, or even mountains. Here we have the principal component analysis of landmark coordinates for the Toyabe and Ruby Mountains. Circles are going to represent individual specimens from these 2 mountains, and these are going to be grouped by species level and fitted with an ellipse. At the Toyabes, our first axis separated specimens very similarly to how we saw in the temporal study, with chrysetids on the left and heteromyids on the right. Chrysetids in that left side indicated by more robustness to the ramus side of the mandible, whereas heteromyids and species on the right side are going to be indicated by larger alveolar regions and more elongated angular processes. At the Rubies Mountains, this heteromyid chrysetid split was inversed, with our lone heteromyid in the upper left corner and our chrysetids clustering at the bottom right corner. At both sites, the lone member of the Dipodidae family, Xapis princeps, clustered close to the chrysetids, and it was differentiated along that second principal component and axis, given its longer coronoid process. The first axis of each site is going to explain about 70 and 50% of the total variation, respectively, largely differentiating between mandibles adept for mastication, our heteromyid sides, and those less so. To better understand those spatial trends in mandible shape, I've taken the first scores along that first principal component and axis and plotted them against elevation. So on the x-axis, we have our elevation in meters, and on the y-axis, we have our principal component scores. And what we see is that in herbivores, there's a very significant trend for increased placement in that first principal component axis with increased elevation. As you move at the Toyabis, at least in principal component space more positively, that's going to be trends towards a mandible shape with larger alveolar robustness and more angular process elongation, both signs of increased levels of mastication. When we look at isotopic niche space ellipse area between our two sites, we see that the Toyabis are going to consistently show higher levels of isotopic niche breadth across most species. Neotoma cinerea was the only species showing a decrease in isotopic niche breadth at the Toyabis when compared to the Rubies, and Xapis princeps was rather stable. When I combined morphological variation measured by principal 1 and 2 scores with the dietary flexibility measured by isotopic niche breadth ellipse area, we don't quite see the significant trends that emerged in our temporal study. And instead, we see these slightly positive trends. It's unclear if this is due to spatial variation not being as strong as in the fossil record, or if this is an example of smaller sample size, or if these two axes of variation truly are decoupled. My last question was assessing the use of space for time substitutions and whether or not the variation a species can exhibit through space today can be used as a proxy for the potential variation a species could exhibit through time. I combined the temporal data from Two-Ledge's chamber with the spatial data from the Ruby and Toyabe mountains to help answer this question. Given that fossil time bins are averaged across much larger intervals in the span of tens to hundreds of years, while modern data is averaged across a week or a month span, I naturally expected to find more variation in the fossil record, given these larger time intervals and a larger opportunity for variation to be captured within an individual. Here, we can see that relationship between morphological variation on the y-axis and isotopic variation or dietary flexibility on the x-axis. And a significant trend emerges across these species through time and space, indicating that with more resources, there are more morphologies to help satisfy all of those different resources. Interestingly, counter to the initial prediction of fossil data points being more variable, given the larger time intervals that they have been averaged over, we see that fossil time period or fossil data points have been mostly clustered in that left side of the graph. Those are indicated by the open circles, whereas the closed circles represent the modern data points. With the exception of one sample that showed high levels of morphological variation and two that showed high levels of isotopic variation, most of the fossil samples fall in this lower left corner. And this is going to indicate a increased sensitivity within our modern species, possibly to environmental change or things like anthropogenic change that are exclusive to the modern. Furthermore, the fact that we see modern specimens with higher levels of morphological variation rather than their or isotopic variation relative than their morphological variation, this is going to suggest that species are responding isotopically more so than morphologically in the modern. When we look across our phylogenetic, dietary, and body size groupings, we see these same trends emerge of clustering of fossil data points on the left side. For our phylogenies of heteromyids and chrysetids, we see these fossil points at lower levels. Additionally, for diet and body size, we see granivores, omnivores, and small-bodied species clustering fossil data points at low levels of variation. Our large-bodied and herbivorous species show high levels of morphological variation in the fossil record. In summary, these two sources of variation, morphological and dietary, are important in species buffering, possibly acting together. Species variation has been repeatedly shown to be higher in the modern than during the past, indicating a possible sensitivity to anthropogenic activity and or warming. And high levels of modern variation is going to invalidate this space for time substitution. I would like to thank everyone that has helped me along this journey, whether it's been providing feedback for my talks, my abstracts, helping with code, or just being a friendly support. I'd also like to thank David, who's been just a great mentor over these last couple of years. And this entire master's project was kind of a brainchild of mine and his. So just very thankful for everyone who has helped on this journey. I'd also like to thank my committee, who has been a constant source of support and guidance over the last couple of years. And I'd really like to thank Becca for taking a chance on me as a student and really letting me show my abilities as a scientist and a researcher, and just all the support and guidance you have given me over the years, and inspiration that you have given me. I'd also like to thank my at-home support system, my 5 dogs, and my partner, Matthias, who has been a constant source of love and support and has made countless figures of small mammals that you guys have seen throughout this presentation. I'd like to thank my funding and the museums and collections I've worked with to help generate this data and research. And if anyone has questions, I'd be happy to answer those now.
Great job, June. I think the best way for questions is if people have questions, they can put them into the chat. Or you can just unmute. Actually, you can't unmute yourself because it's a webinar. So put your questions in the chat.
Thank you guys for all the support. These aren't questions, but they're nice to read.
Wait, Ricardo says you can also use the Q&A prompt for any questions. Thank you.
Okay. Okay, so I've got a question. Do you think those adapted to warm and dry, adapted to warm, dry will increase in abundance, or will others adapt to warm, dry conditions? So specifically heteromyids, a lot of them show increase in abundance in the modern. Species like Chaetodipus formosus have really shown a spike at Two-Ledges Chamber in the modern, and these are very warm and dry adapted critters. So I do think having that predisposition to thrive in warmer and drier conditions will obviously help them. The problem that we see is a lot of these species are granivores, and counter to what we would expect, the granivore lifestyle is not as fruitful as we would hope, given the increased levels of anthropogenic activity and invasive species. So I would think that a species being able to adapt to those warm and dry conditions, but also be able to adjust their diet to maybe incorporate invasive species like cheatgrass would also serve them really well.
Okay, well, if there are any other questions,
I'll take it.
You know how to find June.
Oh.
Oh, wait, hold on.
Got 2. What would you speculate would occur in birds over time? This could be birds that fly or don't. Oof. I would imagine that, things like beak morphology and obviously play a very vital role in buffering bird species through time. And that, their dietary flexibility would also play a vital role. I would imagine similarly that dietary flexibility might be a more immediate response than fully restructuring bills and things like that. And one more, do you think that species who have more dietary flexibility also have more mandibular flexibility, or do they have the perfect mandible for generous diet? It's really hard to quantify that morphological plasticity over an individual's lifetime as opposed to the, generational shape change. But I would think that species that are highly variable morphologically would also exhibit higher levels of mandibular plasticity within their lifetime. We see that with Philip Anderson's morphological plasticity paper that, small mammals, especially more generalist species like mice and rats, have the ability to really restructure their mandibles at fast rates. And one more. Do you think my results apply to other animals in other areas other than the Great Basin? I would like to think that, the research questions that I've posed are, not specific to these exact critters. I think the species that I chose and the study system I looked at were great because they satisfied a lot of the research needs. But I really think the process of morphological versus isotopic assessment could really be utilized across different scales and different sites. Why did different sites not trend the same time? So for the case of temporal sites, that could be chalked up to a lot of different things. These have different local histories. So even though they do span the same time period, they're at different regions. So we see that they have different local climates, they have different levels of anthropogenic activity and different plant cover. As in terms of the spatial sites, the Ruby and Toyabe Mountains, the Ruby Mountains are slightly higher in latitude and also just generally colder and wetter, and they have higher levels of plant diversity. So that could be a great reason as to why we don't see the exact same trends unfolding at those two sites. What is next? I am still figuring that out. The idea of a PhD is interesting. And, having almost completed this, it makes it seem a little less scary. So that's definitely something. I really do like education and outreach and instruction. So I think something in academia is definitely the calling. Birds restructure their skin to change evaporative water loss rates through the integument in hot environments. Is this known for mammals? Don't imagine you can get at this with fossils, maybe skins. I actually don't know if they are able to restructure their skin. I'm sure if they can, that might be something we see in heteromyids that are really, really adapt for hot environments. But I don't know. It would be a very interesting kind of, life history question and could couple nicely with the dietary flexibility side. So sorry for not a better answer than that. What was your favorite part of your project and or whole experience? So I guess it wasn't technically part of the project and it's not my absolute favorite, but one of my favorites was going out to Death Valley and Western Nevada with part of the Gnarly group. That was with Becca and Jesse Laney and Tara Smiley and Catherine Badgley and a bunch of other people that look at small mammals. And really kind of getting to go see some of these areas that are adjacent to what I'm working with, the same systems, and really kind of rekindle that childlike joy that I had, with things like dinosaurs and biology. It really kind of, made me feel like this was the right decision. So that was definitely one of them. And then kind of a cop out, but the friends and relationships I've made along the way, and really kind of getting to know other researchers and just get to know other people.
All right, we should call it there. Thanks everyone for coming and for cheering June on and for your questions and your support. Committee, I sent you an email with a separate Zoom link for our closed door session, and I think we should take at least a 6-minute break. Take longer if you need it, but I will open up that other Zoom room starting at 1, and I will see you all there. Thanks a lot, everyone.
Yeah, thank you guys so much.